Good morning to one and all. I am Dr. P. Ashokkumar, Assistant Professor, Microbiology, Samudra Arts and Science College, T.Gode. Today I am going to talk about parasitology and a gentle introduction about laboratory diagnosis of parasites. Then first thing, what is meant parasitology? It is deal with animal parasites which infects and produce diseases in human beings. So parasite is a living organism which lives in upon another host or organism and take nutrition from directly from host. So it can classified into different groups: protozoa, helminthes, and nematodes. So these are the different classes of parasites, and broadly classified into two groups: ectoparasite and endoparasite. Ectoparasite it lives on the surface of the body of the host without penetrating into the disease. But whereas endoparasite they live within the body of the host, invade into the host tissues, and it will produce some diseases. Then obligate parasites, it cannot live without any host. Then another one, facultative parasite, it can live only in parasitic life or free living life, that is called facultative, either host or without host. Then accidental parasite is this type of parasite lives in unusual host, some accidental host. Then last one, abundant parasite or wandering parasite. These they infect host where they cannot live or develop further development. Then what are the different type of host? Host is an organisms harboring and take the nutrients and provide the nutrients and shelter for the parasite. First one, definitive host. This host in which adult parasite replicates sexually, and it is called as definitive host. Then intermediate host, in this host asexual multiplication takes place in the intermediate host. Then reservoir host, in this host parasites only storing or percentually same take place in over long period, and this reservoir host also sources of infection and absorption. Then parentenic host, in this host the parasite lives but cannot develop further, and it are not essential for life cycle known as parasitic host. Then what are the sources of parasitic infections? Human beings. Man is one of the sources of reservoir for the majority of parasitic infection, example amoebiasis, then enterobiasis. So these are the different sources of parasitic infection. This infection is transferred from one infected man to another infection. This is called anthroponosis. Then second one, animal sources. So some of the animal act as a sources of parasitic infection, that is called zoonotic disease. Some of the animal have some parasitic disease, parasites in host as a normal flora, but it acts as a pathogen for human beings. Then next sources vectors. Vectors usually transmit the disease from one source to another source, that is called vectors such as mosquito, houseflies, then some of the ticks and mice. So these are act as a vectors. Then next one, contaminated soils and water. Soil is one of the major sources for different type of parasitic infection. Soil is polluted with human fecal material containing eggs of parasites or adult worms. So some of the morphological form present in the soils. So it also soil also act as a different sources of parasitic infections. Then next one, raw or undercooked meat. So some of the meat also having some of the parasitic infection. So, it also act as a sources of infection. Other sources like fish, crab and aquatic plants are also sources of infections of parasitic infection. Then modes of transmission. What are the mode? First one, oral or fecal-oral route. So, most of the parasitic infection transmit through the oral or fecal-oral route through contaminated water or contaminated food or vegetables through oral route. Then second one, penetration of the skin and mucous membrane. So, some of the parasite enter into the skin and causes some diseases. Then third one, sexual contact. So, one and only Trichomonas vaginalis is one of the most frequent parasite to be transmitted by the sexual contact. Then next one, bite up vectors. So, some of the vectors also transmit the disease such as malarial parasite transmitted by the female anaphylaxis mosquito, then filariasis transmitted by the culex, then leishmaniasis disease is transmitted by sandfly, Chagas disease is transmitted by redweed bug, then African sleeping sickness is transmitted by test fly. So, these are the some of the vectors also transmit the disease. Then vertical transmission, is to mother to child. So, it is also one of the sources of infection. Then blood transfusion, contaminated blood or infected person plus without any identification transferring blood, it can also disease. Then autoimmune sometimes same persons same persons infected through improper washing or contaminated hand. So, it is also one of the sources of infection, mode of transmission of infection. Then life cycle of parasite. So, most of the parasite are having different life cycles. So, each and every parasite having simple life cycle or in a direct or complicated life cycle. So, most of the parasites requires one host or else two hosts or three hosts. So, it may be depend upon the parasite nature. Then pathogenesis of parasitic disease is different categories. First one, mechanical trauma, then inflammatory reactions, then space occupying lesions, enzymes production by lytic enzymes productions and necrosis, then toxins, some of the toxins were produced, allergic manifestations, then secondary bacterial infection, then neoplasma. So, these are the pathogenic changes in parasitic disease. Then, so most common using laboratory diagnosis of parasitic disease. So, this is most common for all, but each and every parasite having different parasitic identification technique. So, most common technique, first one morphological identification technique, then cultural methods, then immunological methods, then molecular methods, then intradermal skin tests, then genodiagnostic, animal inoculation and imaging techniques. First thing, morphological identification technique. So, parasites can be identified based on the morphology by either macroscopic or microscopic identification. So, macroscopic identification only based on the sample nature and sample quality and and sample nature. Then macroscopic identification is first one majorly used for examination of stool samples. So, stool samples is examined by macroscopic identification first. So, mucoid bloody stool found in acute amoebic dysentery, then intestinal cystomasis and invasive bladder Then dark red stool indicate the upper gastrointestinal bleeding and bright red stool that indicate the lower gastrointestinal infection. Then fourth fourth flea often stool it containing the giardiasis. Then adult worms like roundworm, threadworm and segments are present may be seen in the fecal samples. Then microscopic examination done by the direct microscopic or indirect microscopy. So, first one direct microscopy means using direct wet mount. Direct wet means one drop of saline sample or one drop of leukocytine or some some stainings are used to examine the parasites. So, one drop of saline or water or iodine placed on the glass lid and little amount of stool samples is mixed and directly examined under the microscope in 45x. What is the useful of direct wet mount means? It useful in detection of parasites and cyst of protozoa and eggs and larvae of the helminthes. Motility of trophocytes and larvae can be demonstrated in acute infection. Bile stain are also used to some of the eggs can be identified. It can appear in golden brown and non bile stained eggs are appeared as colorless. Then iodine wet mount it is used to detect cyst, helminthic eggs and larvae. Then it is also helpful in species level identification. What are the disadvantages means? Iodine wet mount is sometimes it will kill the parasitic of parasites and sometimes it can also stop the motility of the some of the parasites. Then bile staining property cannot be applied in iodine wet mount. So, what are the different iodine stain mean? Lucas iodine, B antimony stain, then Dobel's iodine stain. Then permanent stain smears. So, what is the use of permanent stain? It can be used for storage at long time for epidemiological purposes and teaching purposes. Permanent slates can be prepared by using three different methods: iron hematoxylin stain method, trichrome stain method, modified acid stain method. So, these are the methods helpful to diagnose the cyst and trophocytes by the staining their internal structures. The next method, immunological method, that is otherwise called concentrated method. A concentrated method it contains two different, broadly classified into two different methods. One is sedimentation technique, another one is flotation technique. Sedimentation technique furtherly classified into two different techniques: formalin ether concentration technique, then formalin TPAL concentration technique. Then flotation technique it consists of three different methods: saturated salt solution technique, then zinc sulphate flotation technique, then another one is sugar flotation technique. So, these are the concentration method used to concentrate the sample and after concentration these samples are examined under the microscope. Sedimentation technique. So, what is the principle of sedimentation technique? This technique used to sediment the parasites under centrifugal force. So, all the parasite present in the samples using centrifugal force it settle down at the bottom of the centrifuge tube. So, first technique formal ether sedimentation technique. What is the procedure means? About 4 gram of fecal material is transferred into the 10 tube containing 10 ml of 5 to 10% formalin and mixed thoroughly allowed to stand for 30 minutes. Then this mixture is filtered in a 15 ml conical centrifuge tube covered with 2 layer of gauze. About 8 ml of filtrate is collected, 3 to 4 ml of formalin is preserved stool. Then stop. Step 3, next one 0.85% of saline is added to the top of the test tubes and this top of the tube and centrifuged, then supernatant is discarded and again formalin is added and centrifuged for 10 minutes and again sediment is resurfaced in 5 to 10% of formalin and again centrifuge this is the final step to remove the supernatant and collect the sediment, and the sediment is used to examine the parasitic different morphological forms. What is the advantage? Mean this is a sensitivity of detecting ova or cyst increasing by 8 to 10 fold. The size and shape of the parasitic structures are maintained inexpensive, easy to perform. Fecal odor is removed. As a formalin kills the fecal parasite, no risk of occurring laboratory acquired infection. What is the disadvantage? Mean trophocyte forms are killed and hence not detected in this method. Then sedimentation, another technique that is called T.Pal method. T.Pal method is same like from this method we are using formal ether, but in this T.Pal method we are using instead of T.Pal instead of ether we can use T.Pal for the sedimentation technique. The next one, flotation technique, saturated salt solution. In this method mostly used to examine the different type of eggs. So, what are the eggs, parasitic eggs we can examine under the flotation technique. So, first one, salt flotation technique. 4 gram of fecal samples are mixed with 20 ml of saturated salt solution and mixed thoroughly, then allowed to stand for 20 minutes, then it is filtered, then after filtration, then sediments are also mixed with again saturated salt solution and centrifuged, and then after centrifugation, top layer of the tube containing layer containing the different parasites on eggs. What is the advantages and disadvantages? Advantage mainly used to examine the parasitic eggs. What all the reason it is not useful to do not floating egg parasites are we can't examine in salt flotation technique. The next one, zinc sulphate flotation technique. Zinc sulphate flotation in this method 33% of zinc sulphate solution is used to dissolve the parasitic sample, example stool samples, and more or less similar to sugar flotation technique procedures. The next one, egg counting method are also used to identify the different parasitic eggs. Then what are the different type of egg counting method? First one, direct counting method of beaver, then keto selective method, then next one, different type of blood examination. Blood examination, that is called direct wet movement or permanent staining. And direct movement, a drop of blood is collected by finger prick and placed on the glass stick. Our slip is placed over the blood drop and examined under the low objective, useful for detection of microflurae and trypanosomas by their motility. Counting of microflurae may be examined by the blood or Newberg counting chamber. Then examination of blood sample after permanence. So, sometimes we can prepare permanent strain slates and we can examine the parasites. Then next method, examination of blood, quantitative Buffy coat method. So, this method involves preparing blood samples for specialized stain method. You can after centrifugation, the blood Buffy coat layer is collected and exposed to UV rays. So, this will useful to detect the malarial parasite and microfilariae. Then various different methods are also concentrate the blood samples, sedimentation technique, cytocentrifugation, not centrifugation, not concentration, gradient centrifugation, then magnetofluorescence. So, these are the different methods concentrate the blood samples to identify the different parasites. Then next one, examination of skin tissues. Some of the skin tissues are used to examine the different parasitic infection. So, biopsy skin samples are used to examine the different parasites. Then and also examine of CSF fluids are also used to examine the parasites. Then next one, what are the culture media used for the parasitic infections. So, mostly culture media used for studying the pathogenicity, then drug sensitivity, susceptibility test, and preparing antigens for serological test. Culture media used for endomycostolytica, Boak and Dr. Bauhaus medium, then Balamuth medium, Robinson medium, Jones medium, then diamond medium. And for Giardia lamblia, it can be used in diamond medium. Then next one, Trichomonas vaginalis are also used to CPLM medium, that is called cysteine peptone liver maltose medium. Then diamond trypticase yeast maltose medium are also used to cultivate the Trichomonas vaginalis. Then tribulent medium is used for cultivating leishmaniasis and Chagas disease. Then liquid medium for hemoflagellum means leishmania and trypanosoma are also cultured in 600 Drosophila medium with 30% fetal calf serum, and grace insecticide medium are also used. Then culturing for malarial parasite RPMI 164 medium is most widely used method to cultivate the malarial parasite, and Harada Mori filter strip culture, Petri slant culture, charcoal culture. So, these are the different methods used to cultivate the different parasites. The next one, immunological method, that is called based on the antigen and the antibody reactions. Agglomeration test, complement fixation test, gel electrophoresis, indirect hemocclusion assay, counter current immunoelectrophoresis, indirect fluorescent antibody test, ELISA, then rapid immunochromatographic test, then immunoblot and western blot. So, these are the tests used to identify the based on the antigen and antibody reactions. Then molecular methods based on the DNA, DNA or proteins, mainly used to identify the DNA, that is mainly used for DNA probe method and polymerase chain PCR method is one of the most molecular method used for identifying different parasites. Then animal inoculation method, most of the methods are used to identify basic infection, but this animal inoculation method used only for special parasite, or some of the parasites only can be identified by animal inoculation, such as Toxoplasma gondii, then Leishmania donovani and Trypanosoma species, and Trichomonas spiralis. So, these are the parasites can be identified by animal inoculation methods. Then imaging techniques such as X-ray, ultrasound, then computer tomography, then CT, that is called CT scan, then MRI, magnetic resonance imaging technique. So, these are also method used to identify the different parasitic infections. Okay, thank you, one and all.